All right, so the next session is going to be a company overview, company presentation. The company to speak to us is SMSbiotech. Just say a few things about the company. SMSbiotech has a groundbreaking proprietary technology based on its discovery, identification, and naming of smart mobile stem cells. The company is working on a cell therapy-based cure for COPD. Animal studies have shown 100% regeneration in 10 days or less. In laboratory and animal model studies, SMS cells have demonstrated the ability to stimulate lung cells and restore damaged lung air sac structurally and functionally to 100% in 10 days or less. Speaking on behalf of SMSbiotech, very pleased to introduce Dr. Abdulkader Ramo. Dr. Ramo has a bachelor's in biochemistry from the Swiss Federal Institute of Technology in Zurich, PhD in biochemistry from the University of Southern California in Los Angeles, and a postdoc at a major clinical laboratory in Germany. Dr. Ramo is a former assistant professor, has over 30 years of experience in basic and applied research, 11 years primarily focused on SMS cell discovery, development, and technology. Like all of our speakers, he has a much more distinguished and impressive background, but for purposes of getting right into the presentation and devoting as much time to the speaker as we can, I'll just give you the abbreviated version.
And what I would like to talk about today is how we can aspire for longevity through a multi-targeted, orchestrated regeneration that is actually made possible by these small mobile stem cells that we have discovered. Now, if we go towards longevity, I think it's fundamental to understand that there is a big difference between repair versus regeneration. And what we see today as a current modus operandi for medicine is really more like a repair. And with repair, what we have is a very inferior form of tissue being produced through specific drugs and interventions. So what we intend to do and what we should concentrate at is really trying to recover the status of the tissue that is the original healthy status of the tissue that we can put under the auspices of true regeneration. And to do that, we can and we have to promote parenchymal tissue and suppress the fibrotic tissue formation. And this is not trivial, so the question is definitely, how can we do that? Of course, stem cells are the experts really in tissue authentic healing, that is regeneration. This is what keeps us alive, the presence of these stem cells, their proliferation, their differentiation. And when that is optimal, we get optimal regeneration. And the understanding about that is, I think, almost a given. And that's why there has been many cell therapies attempted to treat certain degenerative diseases that has been escaping any solution so far. And I want to mention as a prime example is chronic obstructive pulmonary disease. In that disease, you have the destruction of alveoli, the actual destruction of the lung tissue. Now, when people attempted to use cell therapies such as mesenchymal stem cells mainly, there were many trials doing that, and the conclusion was that adult stem cells, such as mesenchymal stem cells, are quite safe, and that has been actually observed for even a long time, several years after the intervention. However, unfortunately, the use of these cells did not bring about really significant efficacy at all. And there were explanations for that. For example, MSC have low engraftment and they die in large numbers quite early, actually. And the source of the cells sometimes is from the patient itself, and the patient age, so the quality is not so good. And there is also the problem of freezing and thawing these cells, the dose and timing. Patient severity of the disease may be a challenge also to these cells. And then there is a difficult delivery to the lung, which has been a great impediment to the application of cell therapy. There is also very little evidence that mesenchymal stem cells are actually recruited from outside the lung to treat these degenerative cases on a natural basis. So adding mesenchymal stem cells from bone marrow or from fat to the body with the hope to treat this problem in the lung is more like an artificial intervention rather than an augmentation of a natural process. So to overcome these huge obstacles, I think it would behoove on us to look for other cells, and that's what we have done, actually, and we found the SMS cells. These are quite small cells. We're talking about 4 to 5 micrometer in diameter. They're quite transparent. They have escaped detection for several reasons, not just the small size and the transparency, and we can talk about that later. But the important item here is that the fact that these cells present really completely different characteristics than the mesenchymal stem cells, and therefore bear the chance for excellent therapy success. So in general, with cell therapy, we've seen many bottlenecks, and I think we should clearly state those. So there were the problems with safety. I think adult stem cells now are considered quite safe, unlike, for example, embryonic stem cells or iPSCs. Potency is also a concern because some of these cells lose, most of these cells, especially the adult stem cells, lose potency after several passages. Then we have the problem of scalability. We cannot produce large numbers of these cells ex vivo, and that means we have to go back to the donor time and time again. Then there is a problem of targeting. These cells may actually wander around to other locations and not find ever the right target that we intend to treat. Engraftment is, of course, also a problem because the cells simply are not attaching to the right location and therefore do not exercise the needed therapeutic effect locally. And then, of course, the survival. Most of these adult stem cells would die after 2 days. I mean, we're talking about 98% to 99%, unfortunately. So with SMS cells, we're taking care of all these bottlenecks. Actually, the cells are isolated from adult human. without genetic manipulation. And they consider, therefore, adult stem cells. So we consider them quite safe. We tested them a lot, and they turn out to be extremely safe. There is also, in this case, with SMS cells, a multi-targeted orchestrated mode of action that's completely different than any other stem cells in the market, actually. And this mechanism is quite conducive to being efficacious, unlike the other cells. And I will elaborate on that later. Cell biomanufacturing, you can actually proliferate these cells for many passages. We're talking about 27 passages, for example, without loss in potency, which is amazing. But it's actually an inherent feature of a true stem cell, as true stem cells should be able to proliferate and maintain its potency. It's almost a definition of a stem cell. There is also strong selective and direct binding to endogenous cells. So the specific cells where the SMS cells can bind specifically strongly to those cells, something we don't see with, for example, mesenchymal stem cells. The treatment is mainly in endogenous cell therapy. So we don't intend for the SMS cells to differentiate into new tissue because this is actually foreign tissue. And then we will have biomarkers there on the surface that declares these cells to be foreign. What we want here from the SMS cells to stimulate endogenous cells and cause them to regenerate the tissue. So the final tissue is actually patient's own tissue. The cells are also resilient, small, and have low immunogenicity. And I think it makes sense that for our stem cells to be resilient, now these are quite resilient, more than any other cells we actually examined. And I think that's excellent because that helps in the industrial aspect of these cells. And the proliferation, the maintenance, the production schemes get much easier with a resilient cell. In addition to that, this resilience made it possible for the cells to be administered directly to the lung and non-invasively using a nebulizer. So the small size, the resilience of these cells made it possible for them to be administered directly to the lung, which is also unique because there is no cell therapy that can be inhaled, that uses inhalation or nebulization for that purposes. I would like to show some of these characteristics using this video. I hope you can see it. And in this video, we see lung mesenchymal stem cells cultured and the fluorescent SMS cells exiting from a capillary, a glass capillary. These are the small fluorescent green dots that come out and really attach to the area quite next to the capillary because of this strong binding. And once they attach to these mesenchymal stem cells, they actually wander with them around. So what we have here is really an endogenous cell therapy. What we aim at is to stimulate the patient's cells for the treatment, for the purpose of the treatment. And this, of course, this technology is already patented. We received so far 6 patents, and we filed for more, and we look to do more of that, of course, in the near future. Now, the strong binding of SMS cells to mesenchymal stem cells and other cells, such as alveolar type 2 cells, which is quite relevant to the lung because it's a progenitor cell and it is known to repair the air sacs. It's already known to do that. That we see the same strong binding that we've seen with mesenchymal stem cells towards alveolar type 2 cells. And these bindings result not only in stimulation of proliferation, as we have seen with mesenchymal stem cells, but also with a change in gene expression, which is kind of expected. What we didn't expect is that the story is so fitting for healing and treatment of the air sacs and the lung in general. So we see when we analyze the 40 most inhibited, the 40 most inhibited and activated cells, we see countering inflammation, countering fibrosis, countering apoptosis, and countering blood vessel permeation, countering vasoconstriction, which is kind of important for the lung, and countering pathogens, pro-angiogenesis, pro-parenchymal growth, pro-surfactant production, and pro-DNA repair. So when we look at this code here that's being unraveled in terms of gene expression, we can only say that we are decoding a natural event, a natural regeneration that has been induced by SMS cells through the interaction with alveolar type 2 cells. Now, these SMS cells produce also abundant extracellular matrix, and I think it's not a secret if I say that extracellular matrix derived from stem cells have different features than those from other cells, and that some of these features are clearly important for the healing of the tissue. And what we didn't expect is actually the effect of these extracellular matrix on endothelial cells. So when we exposed human endothelial cells to extracellular matrix, we found them to differentiate into small tubuli. Those tubuli are quite stable, actually, for many weeks. After 10 days, the induction goes farther, and these small tubuli form actually large vessels. The endothelial cells form large vessels that can be seen by the naked eye. And this is just a video demonstrating that here we see this larger vessel being formed from endothelial cells as induced by extracellular matrix that has been isolated from SMS cells. Those vessels actually form in vitro in that culture dish, and they end up attaching to the wall of that culture dish. They can be isolated and studied further. But that's not the end of the story. There is more to come. With SMS cells, we observe something I don't think we observe at all with adult stem cells, that their ability to self-organize in 3 dimensions and to build structures in 3 dimensions. So these tiny cells of 4 to 5 micrometer in diameter form actually complex, larger structure without the help of any other cell. So under certain conditions, these cells can form that by themselves. The cells are actually very responsive to molecules that are related to development, that we know that are essential for development. So they have great sensitivity that makes them able to organize. They have the ability to produce extracellular matrix that helps build those structures too. So we end up here with SMS cells having a multimodal mechanism of action. One, if I want to mention the first one, being secreting of matrix and matrix-associated proteins. We've seen those effects on the endothelial cells. By the way, SMS cells bind also to endothelial cells. And we have the selective binding to multiple cells. And those cells are usually known to be important for regeneration. And I want to mention, beside alveolar type 2 cells, beside mesenchymal stem cells, beside endothelial cells, also fibroblasts. So these SMS cells bind also strongly to fibroblasts, but in those cases, they actually inhibit their proliferation. So the other mechanism is about tissue restructuring and remodeling, and I describe some of that in the patents that have been already published and can be visited by interested parties. So what we have here is intercellular induction, intercellular binding, and intercellular morphing. And all that is really a testament to the native intercellular intelligence of these cells. If you are a stem cells, you need to be smart, no question about that. And that's what we mean with smart living drugs. So this native mode of action in tissue regeneration that is based on multi-targeting. And when I say multi-targeting, we target several cells, we target several molecular pathways. So there's multi-targeting that ensures the outcome, the final outcome. But multi-targeting is not enough. You have to have an orchestrated process. This is something where small molecules fail completely. You need to organize all these activities in a way that ensures the final outcome of regeneration. And I think that this is all quintessential to the aspect of rejuvenation. Maybe not that much rejuvenation, but I think you understand the idea. And we give you an example here. That's our main focus. As a small company, unfortunately, we have to focus on one item. And right now we are focusing a lot on the lung emphysema problem, COPD. And here in this case, we used an enzyme, elastase, which is a destructive enzyme that destroys the air sacs, the small alveoli inside the lung that are responsible for the gas exchange, and without which, of course, we couldn't live at all. So these alveoli are destroyed partially using elastase enzyme, and the air sac regeneration was achieved by a single injection of SMS cells via the trachea to the lung. And in 1 week, we were able to achieve actually a structural and functional regeneration of alveoli. That single injection produced actually more than 60% regeneration, but additional doses was able to achieve more than 100% in less than 10 days. And the recovery is not just structural, it's functional. And we tested those animals in a treadmill. We did that because COPD in patient is usually, tested on a 6-minute walk test. And we have colleagues who are quite used to those tests with humans. So we said, okay, the substitute for that would be the treadmill for the animals. These animals were able to recover normal peak velocity and endurance testing after 60% of regeneration of air sacs. So, but I think the important item here is, and that's quite relevant to rejuvenation, I think, is the fact that we were able to have more than 100% regeneration of air sacs using an additional dose of SMS cells. So we went beyond the control. The amount of air sacs density were higher in those animals that were treated compared to the control. So in fact, if you want to take that as such, you can say that we were able to structurally rejuvenate these lungs, and we might actually use that with subclinical cases in humans if we can show clearly that there is no safety concern whatsoever. So currently we are looking for an off-the-shelf COPD allogenic SMS cell therapy. And we are starting our process with a phase 1 clinical study. So we submitted for this clinical trial in Australia, and we are soon to launch our first safety study. We are going to use 3 cohorts with ascending cells amount. Now, these will be 8 mild to moderate COPD patients, and the FDA wants us to look into signs of efficacy even at this stage. And we're happy to do that. Actually, we were quite happy for them to have us select patients instead of healthy volunteers. This is how we're going to do that. As I mentioned before, these cells will be directly administered, and they survive that nebulizing treatment. We've shown that clearly. So the small size and resilience of these cells allows them to be nebulized, and the cells will be inhaled. We expect good engraftment because of the selective and strong binding of SMS cells to lung cells. And then we expect a stimulation of proliferation of the alveolar type 2 cells, of which we know that they produce actually alveolar type 1 cells. So we also expect a change in gene expression. Those changes are quite relevant to the healing of the tissue, to several aspects, for example, like lowering inflammation, reducing fibrosis, and pathogen protection. So these are regeneration critical aspects. Then we also hope for the proliferation of lung mesenchymal stem cells. Now, I have to mention that prior attempts for the lung cell therapy never used lung mesenchymal stem cells because obviously they are very hard to isolate from the patients. I mean, impossible to isolate from the patients, I guess. So what we are going to activate are actually the lung mesenchymal stem cells that are embedded there and are endowed most likely with the function of regeneration of that tissue. We also gonna have ECM produced, and we hope that this will regenerate the alveolar wall and then form, of course, the blood capillary, because without the blood capillary formation, nothing will happen and nothing will succeed. So we have really serious prospects here of succeeding, and I wanted to point out the difference that it makes to choose a different cell, a drastically different cells. And although that seems like discrimination against mesenchymal stem cells, but it shouldn't be conceived like that. We actually view mesenchymal stem cells as very important cells for regeneration. The only difference here is that we actually look for their stimulation inside the body instead of administering these mesenchymal stem cells. And that stimulation can occur through these SMS cells. So in the past, many of these mesenchymal stem cells were used from donors that are elderly, that are aged, to escape the immune reaction and reduce the cell death. That was not achieved quite successfully at all, because there are other aspects that kill the mesenchymal stem cells beside the immunogenic response, and I'm gonna mention those down the line. So SMS cells are derived from healthy donors and used allogeneically, of course. The mechanism of action, contrary to mesenchymal stem cells, SMS cells clearly interact with lung cells. I mean, what is more obvious than a physical strong binding between these cells and these lung cells? And that is, I think, that is a natural phenomenon here. Usually in biology, you rarely see something like that without having an extremely good reason for it. And that's why we are quite optimistic that we can achieve good engraftment and good targeting. We expect, of course, to induce salient gene expression changes, as well as stimulating proliferation of these lung cells. Angiogenesis, we've seen angiogenesis being induced by mesenchymal stem cells, and that's, I think, important, and no question about that. However, when you introduce mesenchymal stem cells, the secretory aspect of that effect is going to be most likely systemic. While in the case of SMS cells, the angiogenesis is localized based on the attachment and the localization of the SMS cells. And that's, I think it's important to be able to localize the effect, even if we talk about, and maybe specifically if we talk about angiogenesis. Now, with regard to cell survival duration, now we still don't have a human data regarding SMS cell survival over there, but there is no question that because of the deficiency of major histocompatibility proteins at the surface of SMS cells, we should expect good survival. We have seen also in animal testing a serious lack in immunogenicity, even with xenogenic exposure. So this is, I think, quite promising. The other thing that is important for the survival of SMS cells is their small size. So they can cross through blood capillaries without problems. With mesenchymal stem cells, they actually get clogged in the capillaries and then get destroyed because of that. So this is one of the major reasons why these mesenchymal stem cells die in large numbers early on after the intervention. There is also specificity. So mesenchymal stem cells of different tissue origins have different features. And those that are tested, as I mentioned, were actually derived from bone marrow or fat tissue, while the fact is what we think is most relevant for the lung is actually the mesenchymal stem cell that is present there. And the mesenchymal stem cells are different, actually. We can assert to the fact that not all mesenchymal stem cells are the same, and they don't respond the same. So SMS cells that are able to bind strongly to those primary lung mesenchymal stem cells gives us a hope to be able to stimulate their proliferation. Scalability, I mentioned that SMS cells can be produced in large quantities. In fact, we produce trillions of these cells without any sacrifice on potency. And that's quintessential if we want to look at industrialization scale stem cells. This is, I think, the answer to that, not those autologous approaches. We providing here an off-the-shelf allogenic treatment that can be revolutionary in terms of the industry access. Now, the route of administration, it cannot be better than being able to route these cells through the lung directly, and that's what we are able to do with these vibration mesh nebulizer that are available in the market. So we didn't create anything new for that. The cells are able to survive this process amazingly and maintain all the potency and all the effects that we have observed in them. Of course, we would like to expand on our platform technology, and there are possibilities for addressing not just chronic diseases like COPD, which has been a challenge for so many decades, but also we might be addressing some rare diseases that are of great concern and for which there is little inducement to treat commercially. We look also into problems regarding blood circulation, inflammation, infections, fibrosis, and internal organs. So we think that SMS cells can provide solutions, and even also in terms of joining other cells. So we could actually think about using several cells in addressing a specific indication or a therapy. We're looking also for stimulating the formation of blood vessels. I think we've shown that in vitro and in vivo clearly the strong effect of these cells. In fact, we know that any regeneration cannot occur without a blood vessel formation. Any serious regeneration requires blood supply, communication between the tissue, and essential for that are the formation of blood vessels. Again, that is the orchestrated approach of the cells that is absolutely essential for creating that complex regeneration process. Okay, and then we have stimulating and inhibiting endogenous cells. And this is really a gentle approach because the cells do not form a new entity here, a new tissue. What they aim here at is really stimulate these endogenous cells and make them ready for regeneration. We have anti-inflammatory effects. There is a plethora of pathways that are anti-inflammatory clearly, not just in alveolar type 2 cells, but also in mesenchymal stem cells. Now, mesenchymal stem cells come in two forms. One is pro-inflammatory, one is counter-inflammatory. And it seems like the SMS cells are switching these cells to the counter-inflammatory format. And that's, I think, is important, definitely during certain interventions. Then there is pathology.
I'm sorry, doctor. We just have about 3 minutes or so.
Oh, okay. Yeah, I'm almost finished. I think, yes, I think I can finish now. So we have pathogen protection, fibrosis reduction, and internal tissue restoration. And thank you very much for your attention.
Okay. Thank you very much, Dr. Ramo. So we do have a few questions. We'll try to get through a couple of them. So you talked about engraftment and the administration is through nebulizer. What percent is lost? What percent of the small stem cells does not graft to the lungs?
Yeah, that's a good question. You see, it depends also on the type of nebulizer. For the one that we are using right now is not a breath actuated nebulizer. So we expect to lose about 70% of the cells. So only 30% will actually go into the lung. And some of those will, of course, be lost to the upper airways, and only a fraction of those will go to the periphery where we need them to be. It's a good question.
And then for the cells that the small stem cells that do reach the target, how long does it take them to reach the target?
Well, that should be quite fast. You know, during the inhalation process, this is something that occurs, you know, almost instantaneously. I mean, in a few seconds, because this is just an inhalation process. Yeah.
Okay. And then you talked about, I think it's intercellular morphing. To what extent does the morphing occur in undesirable ways?
Well, we didn't see that happening at all when we introduced those cells to the animals, and we checked them for a long time, because you know you have to check for carcinogenicity, and you introduce these cells for months actually inside there. So there is nothing of that forming. Apparently, the morphing can occur only under very strict conditions where you have enough of these cells accumulated there, and we don't see it happening in any animals that we have tested.
Okay. And then for what I believe is the impending human trials, I think in Australia with the 18 subjects, is that something that SMSbiotech is doing by itself, or is there a collaboration with another company or a university or teaching hospital?
We collaborate with California Medical Innovation Institute, which is in La Jolla. We did the collaboration mainly on the animal studies, but with the phase 1, we actually hired the CRO. And we have advisors from various institutions like UC San Diego, Professor Peter Wagner, who is a known reference in COPD. And we have also future collaborator will be actually in the US, and we intend to work with them. This is a major hospitals are going to be involved in that.
Okay. And then what's your exit strategy? Is it to license your patents to a large company? Is it to eventually, when the trials are finished, to build your own sales force?
Our exit strategy is mainly licensing, because I think we have a platform technology that can be applied to many indications, and we seek to license every one of these indications, because to pursue all these indications by yourself is almost impossible.
Okay. Okay. And then maybe the last thing, what is your capital position like? Do you have sufficient capital to finish the tests? And at what point would you need to raise additional capital?
Yeah, we are working now on a Series A capital raise. So that Series A is mainly directed towards creating a biomanufacturing site. We want to produce CGMP quality cells at large amounts, large numbers. And for that, we intend to raise about 10 million Series A.
Okay. Well, great. Dr. Ramo, thank you so much for speaking to us and giving us such a thorough overview of SMSbiotech and answering our questions. Very interesting company.